Hey everyone, welcome to week 3 of our PSPirates session. So today we'll be covering lipid metabolism, alcohol metabolism, relevance of genomics, population genomics, and chromosome disorders. So we'll just get straight into it, and I'll pass it off to Claire, who will talk about the first 3 topics.
So I'm starting with lipid metabolism. So we can go next slide. This is just a definition of what lipoproteins are. So they transport hydrophobic lipid molecules in water, and this is because of their structure, because they have a non-polar core, which has the triglycerides inside of it, some cholesterol esters, and some insoluble lipids. Then they also have a polar external coat, which is made up of phospholipids, free cholesterol, and lipoproteins. And as you'll see in the next slide, it's this structure and how much of like each percentage of each is what makes up which lipoprotein. So yeah, and in this diagram, sorry, if we go back one, Emil, sorry. And in this diagram on the right, you'll see this diagram will make more sense, but pretty much if you can look at this diagram by the end of like this week or whenever you do this lecture, and you can understand what's going on here, you've like aced this topic. So yeah, if we go on to the next slide. So how it works is it goes chylomicrons to very low density lipoproteins, to lipoproteins to high density lipoproteins. So chylomicrons are interstitial cells that are after a meal and are approximately 88% triglycerides. They transport triglycerides and cholesterol to body tissues, whereas very low density lipoproteins are made in the liver and are about 50% triglycerides and cholesterol. They transport triglycerides from liver to tissues between meals. And then you have the low density lipoproteins, and these are made in the blood and a majority cholesterol. They transport cholesterol to tissues, and they like class as the bad lipoproteins. This is because you'll learn a bit later down the track how cholesterol can lead to like atherosclerosis. So you don't like want a too high or a too low level of cholesterol. And then you have the high density lipoproteins, and they're made in the liver of a mixture of molecules to reverse cholesterol transport. So they transport from vesicles to the tissues in the liver, and they're classed as the good lipoproteins. And as you move through these lipoproteins, they decrease in size. So I'm not sure if maybe you get a question asked in the exam about 2 different sizes of proteins, you'll know that the smaller one is further down this pathway. And so this is just a summary of, on this slide, it's a bit of information, but of like what happens, how they go to each different lipoprotein. So the cholesterol and triglycerides are packaged into chylomicrons. And if you remember from last week, this like happens in the cells in the gastrointestinal tract. And then these are exported through lymph. And then lipase hydrolyzes the chylomicrons, freeing their fatty acids where needed. And the remainder of the chylomicrons are taken back to the liver to make up the body's cholesterol pool. So then this is where the very low density lipoproteins are processed in the liver, and then they're transported across the body. And the lipase attacks these very low density lipoproteins, freeing the fatty acids. And after the fatty acids have been freed, the low density lipoprotein is then formed. And that's why these low density lipoproteins are formed in the blood. And these go back to the liver and enter via LDL receptors. And then the high density lipoproteins scavenge the body's excess cholesterol and take it back to the liver for excretion. So yeah, now looking specifically at cholesterol, it does not provide energy, but rather it's just a structural component of cell membranes and precursors to synthesis of vitamin D, steroids, and bile acids. It is obtained in the diet or synthesized in the liver, but it cannot be metabolized. So with this cholesterol synthesis, as you can see, it starts with acetyl-CoA. And if we remember, that's like the factor that is needed to enter the Krebs cycle or the citric acid cycle. So it starts with acetyl-CoA, and it is a highly regulated pathway by insulin and glucagon. And HMG-CoA reductase is an enzyme that controls the rate-limiting step and is now targeted by many drugs that focus on reducing cholesterol levels. And if we go next slide. So cholesterol travels in the low-density lipoproteins, and its uptake is via receptor-mediated endocytosis. So as you'll see, like through this, it's just once the low-density lipoprotein binds to the LDL receptor in a cell, that initiates the cell to like enclose it and take it in. And then this LDL is within an endosome, and it fuses with the lysosome, which if you remember from the cells in like week 1, the lysosome has enzymes that break down all the components so that the cell can actually use it. And I just thought these were some high yield points at the end. So like all in all, like they're teaching us all this, but what they're trying to get to is like that cholesterol levels tend to like high cholesterol levels tend to correlate with atherosclerosis. So that's why very high LDL is bad. And then low LDL is also bad because it's negatively associated with heart disease. And then so lipogenesis. So just like a quick note, I know I got a bit confused with this in year 1, but when you read the word, let like the word tell you what's actually going on. So the suffixes help you. So lysis means breakdown. So like glycolysis. So lysis means breakdown, whereas genesis means making or synthesizing. So like lipogenesis or glucogonogenesis. God, I'm really bad at pronouncing it. But yeah, they mean making or synthesizing. And so from this, you can understand that fatty acid synthesis, that lipogenesis is fatty acid synthesis, and it is an anabolic pathway. And so this regulation of fatty acid synthesis is by insulin, and it involves 2 carbon increments, with each increment needing 1 ATP and 2 NADPH. And don't get too caught up on like the NADPH. It's pretty much just the same as NADH or FADH, just another like carrier molecule. So step 1 in this pathway is the committed step. So when acetyl-CoA binds with a bicarbonate ion, it uses 1 ATP, and this is the committed step. So once this has occurred, the whole reaction is essentially occurring. And then malonyl-CoA is converted to the fatty acids by the use of an NADPH, which converts it to an NAD plus, or NADP plus, sorry. And then last, this is kind of like thrown in there at the end. I think you like learn about it in one of your like tutorials or something, and they give you a lot of information, but really, as you can see, like with the high yield point at the bottom, is that overproduction just leads to ketoacidosis, which is a decrease in pH. But for a little bit more information, the ketones are an alternative energy source used by some cells, not liver cells, e.g. the brain or the heart. And acetyl-CoA is used for ketone synthesis when the buildup of acetyl-CoA cannot be used in the citric acid cycle due to a lack of intermediates. So it's made in the liver during starvation from acetyl-CoA for the use in glycolysis. So if you can remember from last week with acetyl-CoA having to go around the Krebs cycle, and going around that Krebs cycle is what produces the electron transport carriers, which then go into chemolysis and ATP synthesis. Except if this acetyl-CoA cannot enter the Krebs cycle because of like a lack of products around the circle, then that causes like the body to go into starvation, and this is what drives the ketone synthesis. So it is like initiated when there's low blood sugar and tissues still need a source of energy. And so like one point they do really like to test you on is that the brain can only use sugars and ketone bodies for energy. And so if there's no sugars, it has to use ketones. And then there's just a point down the bottom that there are 3 types of ketones. So yeah. And then moving on, we'll now look at alcohol metabolism. And just going off the last bit, so I think it'll all make sense when you like get to the great metabolic race, which you might learn about this week, or I think one of our presenters is explaining it next week. But it all starts to make sense then about why we're learning about all these pathways, so that it all fits into like when your body actually needs them. So yeah, going on to alcohol metabolism. Next slide. So in the reaction pathway, ethanol is converted to acetaldehyde by the enzyme alcohol dehydrogenase, and then acetaldehyde is converted to acetate by the enzyme acetaldehyde dehydrogenase. And then acetate is converted to acetyl-CoA, and then, as you know, it goes around the Krebs cycle, or it either goes to fatty acids or it either goes to ketone bodies. So alcohol diffuses quickly across the cell membrane, and absorption depends primarily on the rate of stomach emptying. So on this next slide, just hold in there with me, because this is like a big information dump. And then I've got a summary at the end, and you pretty much, this is explaining it, but at the end, you just need to know 3 points. So the first 2 steps of like like alcohol metabolism, both produce NADH. And this buildup of NADH leads to a lack of NAD+, which is the center of alcohol metabolism. And this is because energy pathways such as glycolysis or beta oxidation requiring NADH slow down. So anaerobic breakdown of pyruvate is favored because it uses NADH to produce NAD+, which is like needed for the Krebs cycle. So this leads to lactic acidosis. And then fatty acids cannot undergo beta oxidation. Instead, they undergo triglyceride synthesis in the liver, leading to fatty liver and cirrhosis. And then toxic components cause inflammation of the gastrointestinal lining, causing poor dietary intake and reduce the ability to absorb thiamine, which is necessary for the conversion of pyruvate to acetyl-CoA. And then hypoglycemia is occurring due to gluconeogenesis not occurring. And so gluconeogenesis, I think you'll learn about next week, but that's just the building glucose to then undergo glycolysis. And then reactive oxygen species are produced, which leads to liver damage. So like it's pretty much all based on that NADH being like that build up, because then there's no NAD+ to allow the normal reactions or metabolism to occur. And so if we go to the next slide, this is all you need to know. So those last points were just explaining it. But if you can like look at this and understand how this is occurring, then like I think you've ticked off the points for alcohol metabolism. But so excessive consumption of alcohol leads to hypoglycemia, lactic acidosis, and fatty liver or hyperlipidemia. So if like I was quickly going to like explain that again, so excessive consumption of alcohol leads to lactic acidosis. This is because alcohol metabolism uses NADH, and so this buildup means that the conversion of pyruvate to lactate is favored, leading to like the buildup of lactate, which leads to lactic acidosis and a decrease in pH. And then because there's no NAD+, the lipids that we ingest are rather converted to triglycerides. And so that's what leads to the fatty liver or hyperlipidemia. So yeah, and then hypoglycemia cannot occur because of these lack of intermediates as well. And then so treatment, they kind of chuck this in on the end, but I think it's pretty low yield. Treatment is with bicarbonate soda or thiamine. And this is a cofactor for enzymes in the citric acid cycle to try and force this reaction to occur and prevent brain damage in a glucose strip line. And then there's also this other thing that your own body does that they teach you about, and it's called the microsomal ethanol oxidizing system. And it's an alternative pathway for ethanol oxidization and is located in the endoplasmic reticulum and is induced by extremely high concentrations of alcohol. The MEOS pathway is an energy-dependent pathway in which NADPH is oxidized to NADP+, and it uses up some of the hydrogen ions produced in alcohol metabolism, which increases pH, and it produces reactive oxygen species damaging the liver and causing cirrhosis. So those two bottom dot points are like the problematic effects of this pathway. But obviously like really high concentrations of alcohol, I suppose we all know is going to be bad for the body, but yeah. So I'll just pass on to Emil now.
Okay, so I'm talking a little bit about genomics and the genomic topics in this week. So the first thing we start off is with the relevance of genomics. So genomics is overall just the study of whole genomes or organisms, and it incorporates these elements from genetics. So the main benefit of genomics is that it offers very personalized medicine. So, it's an approach to treating disease and prevention that can take in your different genes, what different characteristics your family or your family of genes might specifically have. So as a result, there are a few benefits. Firstly, that it can predict your susceptibility to disease. So it can predict whether you're going to be more prone to getting diabetes or more prone to getting heart disease, things like that. And as a result, you can preempt disease progression. Then secondly, there are a few other benefits such as customizing your disease prevention strategy. You can provide more effective drugs if you're specifically targeting a specific problem caused by a specific gene. And you can also control the cost of healthcare because you can reduce the amount of drugs that you might be taking. But the disadvantages is that it can cause unnecessary stress. Then there's privacy issues and ethical issues. So one really big ethical issue with genomics and studying people's genomes is the idea of insurance. So are like people required to disclose the genetic information that they have to their insurance companies? And because of that, are insurance companies allowed to pay extra premiums or charge them extra premiums if they know that someone is more likely to get heart disease or something like that? Okay, so next we talk about single nucleotide polymorphisms. Excuse me. So a single nucleotide polymorphism is a DNA sequence variation occurring when a single nucleotide differs between members of a species. So for a variation to be considered a single nucleotide polymorphism, it must occur in at least 1% of the population. So what this essentially is that if there's a sequence of DNA and it's like ATCG, some proportion of the population might have an ATTG instead of ATCG, and this would be considered a single nucleotide polymorphism. And you can tell that essentially from the name because it's a single nucleotide, so it's one nucleotide polymorphed, so changed into something else. So genome-wide associations then correlate these single nucleotide polymorphisms with common diseases. So for example, one change in a nucleotide sequence could make someone more prone to getting diabetes or having an insulin resistance or something like that. So the Human Genome Project was a project like that went for, I think, 15 years or something like that. And it was aimed to determine the entire human genome and get an idea of what the entire human genome looked like. So pharmacogenetics is then an application of this. So it's the prediction of individual responses to therapeutic drugs using genetic information. So this is based on the idea that certain drugs will work better on certain population and cause certain side effects for others. So I think this diagram here in the bottom right is a really good illustration of that, because it shows you that for some groups, the drug can be toxic but beneficial. And then for other groups, it can be not toxic and not beneficial. And then it can be like not toxic and beneficial. So this is the best group. You want the drugs to have no side effects and be beneficial. But then there can be a group where the drug is toxic, but it's also not beneficial. And that's the last thing that you want. And the issue is that this can occur because of pharmacogenetics and the implications of genetics on the effectiveness of certain drugs. Okay, so now we move on to population genetics, which is slightly different from genomics. And this involves a lot more calculations. So what I'll talk about here is that just the quick difference between population genetics and genomics. So this is fairly low yield, I think, but population genetics is very mathematical. So it involves calculating the prevalence of genetic traits, calculating the frequency of carriers, just passing on traits between generations, whereas population genomics is more of that application of genomic technologies like DNA sequencing and genotyping. So this is the first main calculation that you'll have to do in terms of population genetics, and these are allele frequency calculations. So the first thing I'd say is, even though these are equations, I wouldn't try not to get intimidated by them, because they are actually fairly simple when you realize what it is. So the idea is that alleles are essentially the locuses that you have at genes. So at a certain locus for a gene, you could have 3 possible genotypes. So like the dominant and recessive. So dominant AA, then you have heterozygous big A, little a, and then you'd have the homozygous little a. So what an allele frequency calculation might be is to essentially determine what the frequency of the different alleles are in a population. So we usually call the frequency of the dominant allele P, and we call the frequency of the the recessive allele Q. It doesn't really matter, but I'll show you on the next slide how an example calculation would go. So for example, if there's a population of 100 individuals and the genotypes are 50 homozygous dominant, 30 heterozygous, and 20 homozygous recessive, then the way you calculate allele frequency is the first thing you realize is that the total amount of alleles is going to be 2 times 100, because for every individual, there's going to be 2 alleles at each gene locus, because one big A is going to be one allele and one little a is going to be the other allele. So there's 200 alleles in total. Then what you know is that the total amount of big A alleles, so the dominant alleles, is going to be 2 times 50, because there's 2 here, and then 1 times 30, because there's one big A here. So then you get 130. The amount of recessive alleles will then be 2 times the amount of homozygous recessive individuals, plus 1 times 30, which is the number of heterozygous individuals. So then you get 130 and 70. So you know that there are going to be 70 A alleles out of 200 and 130 big A alleles out of 200. And then essentially you get the frequency from those, 130 on 200, which is 0.65, and the frequency of the little a allele, which is 0.35. So now we get onto genotype frequency calculations. So genotype frequency is the proportion of a genotype in the population. So this can be calculated using essentially just the number of the people with the particular genotype over the total number of individuals. So using the same example that we did before, in a population of 100 individuals, this one is really easy. If it's just 50 AA, 30 A, 50 big A, 30 heterozygous and 20 homozygous recessive, then you'll have just 50 over 100, 0.5, 30 over 100, 0.3, 20 over 100, 0.2. So this is fairly easy, and I don't think you get tested on this that much. Here now, we talk a little bit about the Hardy-Weinberg equilibrium. So the Hardy-Weinberg law states that in large randomly mating populations, allele frequency doesn't actually change over time. Now, this is a fairly, I guess, important concept in that this is describing an ideal population. And this is the reason why there are recessive alleles and dominant traits don't completely overtake recessive alleles, even though they might be dominant. So the expected genotype frequencies, the way the Hardy-Weinberg equilibrium is used, is that you can use the Hardy-Weinberg equilibrium to determine what ideal frequencies of the genotypes or the alleles would be to determine whether a population is in Hardy-Weinberg equilibrium. So the truth is that most populations will not be in Hardy-Weinberg equilibrium, and they'll never be really exact. But if there is a difference from Hardy-Weinberg equilibrium, that can show you that there might be something going wrong or something going differently in the population, for example, like selection pressure or mutations occurring. So for example, if we have a population B where there is 1,000 individuals selected at random, and the 2 alleles at a particular locus, then we have this genotype frequency here. So from here, you can calculate the frequency of allele P, which is what we did before, if we remember, which is 0.7, and the frequency of the recessive allele. Then what we can do is we can use the Hardy-Weinberg equilibrium to determine what the expected numbers of each genotype was supposed to be. So using this equation from the previous slide, this one here, P squared plus 2PQ plus Q squared equals 1, you can use the same numbers of P and Q taken here using the frequency of each allele, and then you can use those to find the expected values of what you would expect to see for each genotype. And here you can see that the genotype frequency of the heterozygous is much higher than expected, which suggests that there might be some sort of heterozygous advantage. And this is the application of the Hardy-Weinberg equilibrium. And as a result, this is how you would use it to figure out something about any population in the wild. Okay, so I mentioned briefly that there are certain things that can cause the Hardy-Weinberg equilibrium to be changed, and these are the main ones. So there's mutations, which can be a source of new allelic genetic variation. There can be migration, so the movement of gametes between populations. Selection, so selection pressures, for example, favoring different genotypes. And the one we talked about before is a really good example of that. because you have perhaps the heterozygous individuals are getting a selective advantage, and as a result, they have a survival advantage. Then there can also be genetic drift and non-random mating. Okay, now to finish off, last topic for today, we're going to talk about chromosome disorders. So chromosome disorders is just a fairly short and simple topic, but it can be like a little bit daunting to look at first. So where we start is with chromosomes. So we learned that chromosomes are the highest level of DNA structure. I think we talked about this last week in that, you have your chromatin, which goes into the chromosomes. And as a result, there is a way of organizing and showing chromosomes to point out where specific genes are on each chromosome. So they're usually shown as an X, which is one chromosome, or as a cinched line, one chromatid. So this would be one chromatid, for example. So the arms of the chromosomes are also named with a certain nomenclature. So the short arm of the chromosome is always called the P arm, and the Q arm is always the long arm of the chromosome. So there's also a method of staining the chromosomes called G-banding, which cause them to have these banded appearances. So these alternating bands of white and black. And as a result, you can actually have a way of pointing out every specific region on any chromosome in a human body or in like an animal by specifically pointing at which arm it's on and which location it's on as well. So for example, here you've got 12p3, which tells you that it's chromosome 12, it's on the P arm and it's band 3. But then furthermore, you can actually go even further in detail, and there can be parts of these bands that people can know cause certain illnesses or certain conditions. So there are 4 main types of chromosomes that you need to know about or that you get taught about. So the first are these telocentric chromosomes, and these all change depending on the location of the centromere. So there are these telocentric chromosomes, there's acrocentric chromosomes, which means that they are very close to the end. There's submetacentric chromosomes, which means they're not in the middle, but close to the middle, move to one side. And there are the metacentric chromosomes. This slide is fairly low yield. I don't think you need to know this too well. Just know that these differences in chromosomes exist. Okay, so now we move on to karyotypes. And karyotypes are fairly important because they're a photomicrograph of chromosomes arranged in succession according to size. So the way that karyotypes are described is they're described by the number of chromosomes, the sex chromosomes, and any abnormalities or descriptions if they are present. So this one here we see is a 46XY normal male, and you see all the chromosomes nicely laid out in order of size, and then the sex chromosomes. So the biggest chromosomes are always first, and then they're organized in terms of size. So they get smaller and smaller until you get to 22, which is the smallest, and then you have your sex chromosomes. So when you describe a karyotype, it's important to know that the way you describe it is just the number of chromosomes. So 46XY, for example, for a normal male, normal female would be 46XX. But then say, for example, there was an extra chromosome 21 here, then that would be 47XX, but then plus 21, which would be a female with Down syndrome. So it is possible for you to get an exam question that shows you a karyotype with some sort of abnormality, and you might be asked to show or click an answer that describes what that karyotype is and what the condition or disorder associated with it might be. Okay, so next we talk a little bit about numerical abnormalities that can occur in chromosomes. So the first main one that we talk about is aneuploidy. So this is when you have a missing chromosome or an additional chromosome. So Down syndrome forms under this section of aneuploidy. So there are a few types of aneuploidy. There's monosomy, which is a loss of a chromosome. So an example of this would be 45XY, where you've lost the 14th chromosome. And these are often lethal, but an exception to this is 45X, which we'll talk about a little bit later. Then there is also trisomy. So this is one that is much more common and it usually can not be lethal. So it can actually, babies can survive with trisomies. And a really good example is Down syndrome, as we've talked about already, and you probably already know about. So often there are very serious mortality impacts, but it's not always lethal. And we'll talk about a few examples of trisomy that can occur where it is not lethal. Now, then there's also tetrasomy, which is extremely rare. But it's not linked to like any major name disorders because it's so rare. And this one is also usually lethal when it happens. Then finally, in terms of numerical abnormalities, there is polyploidy. So polyploidy is different from aneuploidy in that it is a multiple of a haploid number. So instead of having 46 chromosomes, you might have 69 chromosomes and 3 sex chromosomes or 80 something as well. So it's lethal in most cases, but it's normal in things like strawberries, I believe, and like random fruits. This can actually happen. And all of these usually result from a failure during anaphase. This is probably something that you'll go through in depth in a workshop, but I think I've included a slide here that shows what can happen. So usually what happens in meiosis is that the gametes would split normally and you'd have 2 chromosomes on this side and 2 chromosomes on this side. But what can happen is actually non-disjunction. And as a result, one of the gametes gets 2 chromosomes. One of the pre-gametes gets 2 chromatids, and then the other gamete gets 3 chromatids. And as a result, the gametes end up having 3, 3, and then 1, 1. And as a result of this, you can have these abnormalities that occur. So now there are also these structural abnormalities. So there's a difference between structural abnormalities and numerical abnormalities. So these numerical abnormalities are the addition of a whole chromosome, whereas these structural abnormalities are issues within chromosomes. So within chromosomes, there are 3 main issues that can occur. So the first is duplication, the next is deletion, and the final one is inversion. So what can happen is an area of a chromosome, so in this one, the B region, can actually duplicate during either mitosis or meiosis. And as a result, you can get this duplication. Furthermore, you can also get these deletions. So here the B has been deleted again. And also you can get these inversions where the order has been switched around. So as it says on this slide, the arrangements can be balanced or unbalanced. And in an unbalanced arrangement, there is a net loss or gain of genetic material. So duplication here is a gain, deletion here is a loss. So these would be unbalanced, whereas inversion would usually be balanced. So in an unbalanced arrangement, there are usually fairly serious consequences for the person who might have this unbalanced arrangement. Whereas in a balanced arrangement, there might be no net loss or gain, but future offspring who might have this in their gametes can have an unbalanced arrangement because of the other person's sex chromosomes not having this unbalanced arrangement. So under structural abnormalities, there is also something called translocation. And there are 3 main types of translocation that you can know about. So the first one is reciprocal, where 2 chromosomes break and the broken pieces are actually exchanged before reattachment. So here you've got this chromosome, the blue one and the red one, and the blue and red areas switch, and this is called a reciprocal translocation. There can also be an insertional translocation. So this occurs when you have this blue chromosome here, a section is taken out and it's added into this red chromosome. And then finally, you can have these Robertsonian translocations. And this is when, as we talked about before, there are these acrocentric chromosomes. They can lose their short arms and actually fuse, forming one chromosome. So this one is fairly interesting because this is actually a cause of Down syndrome, where this could be 21 chromosomes, and the acrocentric ends of these chromosomes are cut off, and there is this chromosome, one chromosome that contains twice as much of the genetic material, and there will be another normal 21 chromosome, meaning that there would be 1, 2, and then a 3 Robertsonian Down syndrome complication. So now we start to talk about autosomal disorders, and this is one where you can get tested on this quite a bit. So there are 4 main autosomal disorders that you can get tested on. So the first is Patau's, which is trisomy 13. So this is where you have an extra 13 chromosome. And this one has a 10% survival rate to 1 year and has very serious developmental effects. The next one is Edwards syndrome, which is trisomy 18, where you have an extra 18 chromosome. This one also has very serious developmental effects, and not many babies survive. However, one that you all probably know about is Down syndrome, which is a trisomy 21. And this one is much more survivable. And a nice way to realize why this is more survivable is thinking about the size of the chromosomes. So the bigger chromosomes are more likely to have more genetic material. And as a result, losing a bigger chromosome is more likely to result in a greater loss of genetic material. So a nice way to, I guess, maybe not nice, but a nice way to remember this, the order and the chromosomes, is this quick phrase: pedophiles start young, 13, 18, 21. So you have Patau, which is trisomy 13, Edwards, trisomy 18, and Down's syndrome, which is trisomy 21. Now, the last autosomal disorder here is Cri du Chat, and this one is a 5p deletion. So this one is one that we talked about before, where possibly a part of a chromosome can be missing, and portion of chromosome 5 is missing, and it's called Cri du Chat because it means cry of a cat, I think, or cry like a cat. And as a result of this deletion, the infant's cry sounds like a cat, and there are some developmental effects as well. Now, finishing off quickly now, there's these sex-linked disorders. So these are the two main ones here, Klinefelter's and Turner's. So Klinefelter's syndrome is an added X chromosome. So it's often unnoticed because the symptoms can be mild, and the patient is male but has feminine features, infertility, and learning difficulties. This one is usually treated with testosterone, and what can happen is the person with Klinefelter's can develop breasts and have other complications as well. Turner syndrome is a missing X chromosome, which is one that we would discuss before, which is a monosomy. So you have monosomy of the X chromosome. The patient is female, but has underdeveloped secondary sexual characteristics and redundant neck skin and short stature. And this is usually treated with growth hormone and estrogen. And that's the end of our session today. So thank you all very much for coming. And we hope you learned something from what we talked about today. And if you have any questions, of course, feel free to stay behind or pop them in the chat.